Our experiment shows the effects of nitrogen preference of nitrate compared to ammonium on the growth of phyllanthus pollutants commonly known as red root floaters.
Nitrogen pollution is among the most prevalent water problems in growing nations and leads to degraded water quality, collapsed nutrient cycles, decline in biodiversity, and makes previously clean water unsafe for consumption. Phytoremediation, the use of plants to remove contaminants and pollutants. Nitrogen, in our case, has been receiving increased attention from researchers due to its low cost and high efficiency, with a growing body of evidence proving its efficacy. From our research, we sought to determine the phytoremediation suitability of phyllanthus pollutants to contribute to improving phytoremediation systems. Nitrogen preference has strong implications regarding phytoremediation because ammonium is the main nitrogen source in wastewater, suggesting that plants with an ammonium preference will be better suited for phytoremediation. Additionally, previous research suggests growth rate, a measurement in our study, to be the strongest predictor of nitrogen uptake and assimilation. Floating plants are often held in high acclaim in the phytoremediation space due to the known high growth rate and resilience of most floating plant species, in large due to their ability to utilize atmospheric CO2. An example of this can be seen with duckweed, a morphologically similar plant to Phyllanthus fluens, and is considered a gold standard of phytoremediation. Additionally, when exploring the literature on phytoremediation, you tend to come across a striking pattern in which most plants suitable for phytoremediation are often the ones most invasive to non-native ecosystem. So much so that some researchers will cite reports of invasion as an indicator of phytoremediation suitability. However, the reasoning behind this is quite simple. Plants that are invasive tend to be both resilient and fast-growing, two traits virtually ubiquitous in determining suitability. Reports have surfaced of Phyllanthus fluens spreading to natural bodies of water, mainly in Florida, where environmentalists fear that its high growth rate could cause ecological damage. Its invasive potential, coupled with the known physiology of floating plants, led us to choose Phyllanthus fluens as a subject of this study.
This leads us to the research question: what are the effects of nitrogen preference of nitrate compared to ammonium on the growth of phyllanthus fluens, with the end purpose of determining its phytoremediation suitability for implementation in a larger scale? Based on previous research on duckweed, a morphologically similar plant to phyllanthus fluens, we hypothesized that it would exhibit the highest growth using a 3:1 ammonium to nitrate ratio. To confirm this theory, we had two trials. Trial 1 lasted 28 days with 6 samples per treatment. The treatments included so nitrate, a 1:3 ratio, a 1:1 control, 3:1, and so ammonium. These ratios were made using the stock solution shown in the table above. Measurements were taken every Monday, Wednesday, and Friday, including fresh weight with analytical balance for precision, root length in centimeters, leaf amount counting, noting damage that occurred to the leaves while taking measurements, its condition of the leaves, or other factors such as growing buds, survival number, and dry weight at the end of the trial. However, due to the high mortality rate, which could have affected the interpretation of the results, the second trial we implemented various changes, decreasing the medium hardness time span of the trial to 24 days, increasing, increasing sample size, decreasing the measurement frequency, and the addition of a new control, which was water.
All data gathered was then input into Microsoft Excel, where we were able to make the line and bar graphs. One-way ANOVA followed by Tukey HSD was then performed where a p-value less than 0.05 was considered significant. For all trial 1 graphs, the standard deviation was used as error bars, while for all trial 2 graphs, the standard error of mean was used as error bars. All figures are numbered as they are in the research paper, and for all graphs, each colored column represents a different treatment group. These graphs show the total fresh weight for trial 1 of the study. Graph A shows the total fresh weight from day 0 to day 28, and graph B shows the total fresh weight specifically on day 0, day 14, and day 28, with error bars representing standard deviation. The overall trend for all groups other than the sole nitrate group showed that the total fresh weight decreased. However, the total fresh weight stayed similar throughout all days for the sole nitrate group.
Another form of measurement taken was images of the plants for photo documentation of changes that couldn't be expressed through solid data. These images were taken weekly. Here we could see the changes as the surface area significantly decreased from the start to the end of the trial, with algae growth showing in some of the treatments as well as the condition of the plants worsening as the trial progressed.
The figure for the dry weight of the plants for all treatment groups can be seen on the left. The sole nitrate treatment group had the greatest dry weight, which the sole ammonium treatment group followed. On the right, the survival number from day 0 to day 28 is depicted. Similar to previous trends, the nitrate group maintained a survival percentage of 100%, while all other treatment groups decreased in sample size. The sole ammonium group decreased in survival percentage by 50% from day 0 to the last day. The following graphs will portray the total fresh weight, images of plants, dry weight, and survival number for trial 2 of the study. These graphs specifically show the total fresh weight for trial 2, where graph A shows a line graph of the process and graph B shows the total fresh weight, specifically on day 0, day 14, and day 24. It can be seen that the sole nitrate group almost tripled in the total fresh weight, with significant differences seen between day 0 to day 14 and day 24. All other treatment groups decreased in their total fresh weight without any significant differences between them.
In contrast to trial 1, we could see that the surface area is comparatively bigger at the end of the trial, which could be due to a lot of the changes we made for trial 2, as the other factors in the environment were controlled, such as the amount of sunlight. The condition of the plants also seemed to be better, especially with sole nitrate in this trial.
Again, the graph on the left shows the dry weight of trial 2 at the end of the study, and the graph on the right shows the survival number. The trend for the sole nitrate group's dry weight was similar to trial 1's trial 1's sole nitrate group, where they had the highest dry weight at the end of the study. The other treatment groups had a lower dry weight than the control group, with the ammonium treatment group dying off completely. For the survival number, the nitrate group increased in survival number by almost 150%, and it can be seen that the ammonium group died off completely near the end of the study. Compared to day 0, it can be seen that all other treatment groups decreased in survival numbers.
Using past research, we believe that the addition of cecum equilibrium, something that was significantly reduced in the second trial, had significant influence on the results of trial 1. Cecum equilibrium is a general hardness booster with the original purpose of providing calcium and magnesium to the plant. Unbeknownst to us at the start of the first trial, Phalanthus fluens likely exhibited a strong intolerance to water hardness, and that resulted in the high mortality rate of trial 1. Additionally, as seen with the figures described previously, trial 1's results on the ammonium group conflicted with that of trial 2's, with the ammonium group in trial 1 performing second to best, but fully dying out in trial 2. We believe that this was due to ammonium toxicity being alleviated by the high potassium content in cecum equilibrium. As shown in a recent study by Fang and colleagues, potassium dramatically decreased the symptoms of ammonium toxicity in rice seedlings, a plant known to be intolerant to high concentrations of ammonium. To conclude, Phalanthus fluens is a poor candidate for phytoremediation due to its strong preference for nitrate, ammonium intolerance, water hardness sensitivity, and poor physiological responses to environmental stress. However, in optimal conditions, Phalanthus fluens has the potential to become highly invasive due to its high growth rate. Since Phalanthus fluens is unlikely to become invasive in the bodies of water with moderate or high levels of water hardness and low pH, where nitrification is starkly reduced or eliminated entirely, and highly invasive in the contrary, environmentalists should extrapolate the findings of this study to predict and prevent possible Phalanthus fluen invasions in the future. Lastly, our research cements nitrogen preference as an essential consideration when considering phytoremediation, as shown with the sharp contrast between experimental groups. Thank you for listening to our presentation.